Hello, my name is Munya Hamadi. I am an undergraduate from Dr. Ren Guerra's sleep lab at George Mason University in Virginia. My ongoing project is looking at novel sleep-related genes in the Drosophila melanogaster through various sleep trials that analyzes total sleep trends and sleep bouts. For the past 2 semesters, I've been focusing on the straightjacket gene. We chose straightjacket as the first gene of study due to the paper published by Joshi et al. In 2019. They were analyzing the ortholog of straightjacket, known as CA-CNA2D3. This gene is a calcium channel protein that they found to interact with various circadian genes in knockout mice. Some of these genes includes period 1, clock, BMAL1, and NALCN. Thus, we hypothesize that this ortholog interacts with the mice's circadian rhythm. We determined the ortholog of CA-CNA2D3 in flies through the Protein Blast website, where we analyzed protein sequences to determine the fly protein that most closely resembled that of the mice. And since straightjacket is also a voltage-gated calcium channel found in neuronal tissues of Drosophila melanogaster, we believe it is the top candidate to represent the CA-CNA2D3 in flies. The knockout mice were deemed the top candidate gene for sleep effects. However, the way in which it is so in the Joshi paper is not specified. There are also no other papers that we have found discussing specifically the sleep effects of this calcium channel protein. Thus, since the ortholog has been shown to acquire interactions with vital circadian genes, it is expected that there will be some alteration in circadian rhythm in the Drosophila melanogaster with a modified straightjacket gene. Thus, our hypothesis states, if the voltage-gated calcium channel protein, straightjacket, interacts with vital genes for the fly circadian rhythm, then there will be an abnormal sleep effect that will occur through their circadian rhythm. The flies were kept in 12-12 light-dark conditions at 22 degrees Celsius in vials with cornmeal, molasses, and a yeast mixture 1 week prior to running. Total sleep was recorded in Drosophila activity monitors made by Tri-Kinetics Incorporated. These monitors were placed in a 22 degrees Celsius incubator for a week, each holding a max of 32 flies per monitor. The genotypes utilized for straightjacket manipulation includes a GAL4 that is expressed throughout all nervous tissue when activated, which is the 8765, that can either activate the 25807, which is a UAS trip, which decreases the amount of straightjacket RNA being produced, or an MIC mutant, 34109. To obtain the activated genomes in the flies, we crossed a GAL4 with the UAS genotype flies, as well as the mutant ones. Only virgin females were utilized, females that were within 8 hours of eclosion, as well as just any kind of male. We had a total of 4 different genotypes of offspring, one where the male was the 8765, which was crossed with either a virgin female, which was the 25807, or a virgin female 34109, and vice versa. This was to ensure that any abnormal phenotypical response were not determined by the sex of the parents. This would indicate that straightjacket were associated with a sex chromosome, thus ending us with 4 different genotypes of progeny. Data analysis was done with the DAM2 software and a macro Excel sheet created by Paul Shaw. The Excel sheets were used to organize the data points on the average beam crosses per fly from ZT0 to ZT23. The total sleep was graphed with RStudio. These graphs are showing the total sleep of the flies for 4 days after 2 days of allowing the flies to readjust to their new environments. The N value can be found under these figures. I want to strictly emphasize that this is strictly preliminary data, since statistics have yet to be run on total sleep. However, qualitatively, the female flies of the offsprings, more specifically 8765 female x 25807 male, which is this blue line here, which is the UAS GAL4 system, have decreased sleep at a phase change from light to dark, which is ZT11. Qualitatively, there is no substantial difference for the males, as shown with this graph here. Preliminary data of the straightjacket mutated flies shows an increase in sleep during the light phase for both of the crossed progeny, which is shown in green and blue, when compared to the parental 34109, which is shown in red. However, there is a decrease in sleep when compared to the GAL4 8765, which is in purple. The females have a more distinct difference qualitatively than the males. These N values can be found under the figures. Once more, I am Munya Hamadi. I am an undergraduate student studying at Dr. Ren Guerrero's Sleep Laboratory at George Mason University. As you can see to the right, the laboratory consists of Dr. Guerrero as the principal investigator and the other undergraduate student, Matthew Perez. I would like to thank George Mason's Oscar URSP program for funding me over the summer, as well as the Oscar Undergraduate Student Travel Fund for providing me the financial aid to attend the conference, as well as the Interdisciplinary Program in Neuroscience at George Mason for providing me with the proper resources to run these experiments. Please reach out if you have any questions or comments about this project.